Hey everyone, and welcome to the continuation of the Investing in Cure Summit. I'm Ben Yerxa, CEO for Foundation Fighting Blindness and also the RD Fund. I'm joined by our co-moderator, Eugene, a physician scientist and serial inventor and entrepreneur who is vice chair at Foresight Labs and is a member of the RD Fund board of directors. Gene, thanks for joining us. First, a few quick housekeeping items. Closed captioning is available for today's session. You can turn on the captioning by selecting the CC icon in the bottom right corner of the video window. You may also ask questions during today's session, so you can submit your questions through the chat and the Q&A modules that are located to the right of the video player. Also, all features are available by tabbing through and then selecting by pressing Enter on your keyboard. So, all right, let's get started. Today on the ecosystem panel, we really want to tackle a variety of topics that bear down on the central question of how do we actually get this job done, which is bringing treatments and cures to those with vision loss due to retinal degenerations. We have 7 panelists today representing some, but not all, of the sectors that are needed to work together, sort of like a village, if you want to think about it that way, to accomplish the Foundation's objective. So let's start first with some quick introductions. We'll have each panelist state their role in the ecosystem and connection to the Foundation. And if we can go ahead and get started with Richard, if you want to go ahead and kick us off.
Sure. Thank you, Ben, and thank you for having me here today. My name is Dr. Richard Awdeh. I'm trained as an ophthalmologist, went to medical school at Yale, completed my training at the Memorial Sloan Kettering Cancer Center at Duke University. I moved on to the faculty at Bascom Palmer for over the past 10 years, where I ran an NIH lab focused on nanoparticle drug delivery to the back of the eye. And currently, I'm CEO of a healthcare technology company called CheckedUp that works with many subspecialties in the U.S., providing large format electronic displays in office and exam rooms.
Great, thank you. Next, Kevin Bitterman, please.
Hi, everyone. Thank you for having me here. Kevin Bitterman. I'm a partner with Atlas Venture. Atlas is a Cambridge, Massachusetts-based venture capital and company formation firm. We've been around for about 25 years doing early stage biotech investing. Our bread and butter is taking technology out of academic universities or elsewhere, incubating that technology in our offices with a very talented group of entrepreneurs and drug hunters and building companies. And we do that across therapeutic areas and across drug modalities and been quite active in the ophthalmology space over the years. My background, I've been at the firm for about 4 years. I spent 13 years at another Boston-based venture firm prior to that, and have a PhD in genetics from Harvard Medical School. Looking forward to the discussion.
All right, great. Thanks, Kevin. Next up, Stéphane.
Thank you, Ben. Thank you for having me today. So my name is Stéphane Boissel. I'm the CEO of SparingVision, which is a very happy investee company of the RD Fund from the Foundation. SparingVision is a genomic medicine company that is only involved in ophthalmology. Our lead asset is the first gene mutation or mutation agnostic, gene therapy to be going into in-man trial. We expect to be starting that trial later in 2021. And next to SPVN06, we are building what we believe is an unparalleled portfolio of genomic medicine in the space. Thank you again for having me today.
All right, thank you, Stéphane. And going around the horn, next is Wiley Chambers.
Good morning or afternoon, depending on where you are. Thank you very much for having me. My name is Wiley Chambers. I am the director for the Division of Ophthalmology in the Center for Drug Evaluation and Research at the Food and Drug Administration. I have been at the FDA for 33 years, always reviewing ophthalmology products, but sometimes reviewing other areas. And I'm very pleased to be here. Thank you.
All right, thank you, Dr. Chambers. Laura Manfre, you're up next.
All right, thanks, Ben. I'm Laura Manfre, and during the day, I'm a director of learning and development for Hewlett-Packard Enterprise. And then evenings and weekends, I am the co-founder and board chair of Sofia Sees Hope, an LCA patient advocacy organization. And all the time, there's no getting out of this, I am the mother of 3 children, ages 10 to 20, and my middle child, who is now 18, is affected with LCA.
Great, thanks for joining us, Laura. Steve.
Great. First, thanks, Ben and Gene, for the invite to participate. So I'm Steve Pakola. I'm the Chief Medical Officer for REGENXBIO, which is a gene therapy focused clinical stage company. And our mission is to improve lives through the curative potential of gene therapy based on our AAV, adeno-associated virus vectors. And the company's actually been at this for over 10 years. So really from back in the days of where it was a little quiet in gene therapy. And obviously there's been a lot of advances over that time frame. So I joined the company 2 years ago based on my background in drug development in ophthalmology, and most of that within retina. And interestingly, our lead program is a local retinal gene therapy delivery program, but not for an inherited retinal disease, but rather use of gene therapy to deliver a therapeutic protein targeting a well-validated target for more common disorders. But I joined based on that, but also our platform, which has the opportunity to treat monogenic disorders, including inherited retinal diseases, but also diseases outside the eye. And so I guess I'd say my role here is from the drug developer standpoint. And I think ecosystem is a good, really apt phrase here, where it does take a village from all these different angles. And I've kind of seen working at REGENXBIO, when you're involved in the rare monogenic disorders, how critical it is to involve all the different parties within the ecosystem. So it's really great to see a panel like this that has so many of these different voices that are important.
Awesome, thank you, Steve. Last but not least, Tarran, you're up.
Hi, this is Tarran Pierfelice. I'm the business head of the gene therapy franchise at National Resilience. We go by Resilience. National Resilience is a about 9-month-old startup that's focusing on investing and developing innovative manufacturing solutions and technologies and platforms to really help better manufacture complex medicines in the cell and gene therapy space. Our gene therapy franchise focuses on viral vectors, a big emphasis on finding better ways to manufacture AAV and other viral vectors that are crucial for curing debilitating diseases in the cell and gene therapy space. We're really just trying to find better ways to make these medicines more accessible by having better productivity, but also making these medicines commercially viable by finding better ways to manufacture them at scale. My background includes being a stem cell biologist by training. I studied at the Johns Hopkins University School of Medicine, and I've spent about 10 years in various business development roles, both at gene therapy manufacturing organizations called Paragon Bioservices, which is now part of Catalent. I've also spent several roles in business development at innovator companies in the cell and gene therapy space, most recently at Editas Medicine, where I was leading the business development efforts in the cell therapy pipeline. Nice to meet you.
All right, great. So now I will hand it over to Gene to kick us off with the first question.
Thank you, Ben. Wiley, always kind of one of the stumbling blocks that we have is understanding, how to get a new therapy approved. I mean, there's and then retinal degenerations. For lots of reasons, we've had problems with getting and identifying the most relevant early clinical endpoints that would allow us to get something approved. We would love to get your thoughts. I know this has been an ongoing decades of conversations. Just help us here. Wiley, do you have how often do people kind of come in and make requests or that seem kind of off the mark? They say, look, we just need to do this. This is going to work. Can't we determine efficacy later? We've got safety or something like that. What are the mistakes that people do most commonly, you think?
So they don't ask us those kind of things more than twice a day. Everybody would like to have products that are safe and efficacious out immediately. The effort that it takes to develop a product frequently requires believing so much in that product that sometimes you have the objectivity and sometimes you don't. We're not invested in any one particular product. We may seem cold to the development, but that's not really what we're trying to do. We're trying to make sure that if somebody uses a product that's approved, they can be assured that they're going to get a product that does what it says it's going to do. And if that means holding something up until the product has been shown to do what it says it does, so be it. That's the way Congress wrote the law.
Ben, how do you feel about working with the FDA? You do a lot of, as being a patient representative organization and driving these things. How do you see that? Are we doing a good enough job?
Well, yeah, we appreciate the partnership with the agency. Obviously, we are covering new ground here. Eye charts are not going to do the job, for most of the things we're working on. So we just appreciate the partnership with the agency. We tend to be sort of, a middleman between industry and the agency, and to the extent we can facilitate, better conversations around how to execute on these things. We're all about that. And so I think that's where some of the action is really getting the right people around the room to tackle these big problems and then have an action plan to do it. Well, good. Maybe Steve, did you want to chime in?
Sure. Yeah. So like you, Ben, we've collaborated over the years. So I'm sure you agree with this as well. I've worked in ophthalmology and other therapeutic areas previously and now, and hearing Dr. Chambers talk about the goal to benefit patients, I think it is important to say how fortunate we are in ophthalmology and with the FFB and in general where it really is a very constructive dialogue in how to advance to benefit patients in ophthalmic products. So I think we're really in a good position. So I can only second that it's really not cold at all. Even though we on the sponsor side ask once a day at least for different endpoints, it's always a very logical, objective, balanced way of how do we get safe and efficacious medications to or whatever therapy to the patients that need it. I will say one thing I've realized, having traditionally not worked in rare diseases, but in the last couple years, when you work in rare diseases, there's that sense of urgency becomes very guttural almost, because you become very connected to families and patients. But yet you, even more so, you return to the roots of the concepts of safe and efficacious, that you need to be urgent, but you also want to do it the right way to make sure that it gets to the right patients and can have the most benefit for the patients.
Yeah, that makes a lot of sense. Thanks, Steve. Why don't we move on to another topic, which sometimes we don't talk about as much. We talk a lot about animal models and things like that, but manufacturing. And Tarran, I'd be interested in your take on manufacturing now, knowing what we know now about cell and gene therapy. I was impressed to see the latest figures. So there's something like 3,400 programs in development in cell and gene therapy right now. So why resilience? Why innovation in manufacturing? Could you speak to that?
Absolutely. There's been a disconnect in terms of the speed with which innovation in products is moving very, very fast, but the speed with which innovation in manufacturing hasn't really kept pace with the speed that products have been developed. And it's created this incentive to move very, very fast to the clinic and not putting an upfront investment in manufacturing to have better, more commercially viable manufacturing solutions. I've seen over the last decade, working with both at innovator companies and manufacturers, people wanting speed to the clinic to get proof of concept and not really spending the time on developing commercially viable platforms to manufacture that's going to be able to scale and bring down COGS. People are using sometimes 20, 30-year-old processes that are really old and outdated. And so what the industry is really yearning for is better manufacturing solutions that provide better, higher yields, 10 to 100-fold higher yields, faster ramp up to GMP in order to drive down COGS over time. Also improving downstream in the analytics that go along with the manufacturing to increase yields and purity. And so what has been good enough is not good enough for the future for these cell and gene therapies to extend to patients in the rare disease space, but also as innovator companies move into more complex diseases that have larger patient populations. And so what several companies in the manufacturing space, including Resilience, are trying to invest upfront on these manufacturing solutions so that we can offer options to scale, bring down COGS, and have better product characterization to improve safety. And ultimately, this is all for the patient. We want to get things that are safe, scalable, and affordable to patients. So I'll stop there.
I have a follow-up question for Tarran. From the, we identify, say, it works in our animal model, but what does it take once we have a construct and we're going, we hand it over to you to get through the manufacturing, preclinical validation, and into the first patient? What's the time scale there? And
So the time scale, there's always pressure to get to clinic as fast as possible, right? And you might have seen, because of that pressure to move fast, there's been a lot of issues actually with the CMC section in the IND application that you've seen with many of the companies out there, also associated with variability in manufacturing. But traditionally, the timelines of getting a construct or something that's maybe a bench scale process to GMP, phase 1 material release can be anywhere from 9 months to a year. And that really is not fast enough. And so what ideally we want to move faster, right? And ideally we want to shorten those timelines to less than 6 months. And it's going to take an investment, not just in the biology, but it's also going to take an investment in the digital infrastructures that can help find efficiencies of moving products through process development, analytical development, and then scale up into GMP and release. But ultimately, the faster, the better, but never, ever compromising quality.
Thanks. Maybe I'll take the next one, and I'll direct this to Kevin. The source of almost all things is the resources to kind of get these teams and ideas properly funded. How do you see that? How does that really work? And do you have some insights for the ecosystem?
Sure. Well, I'll echo what was said before, that ecosystem is the right word, and it certainly does require a village. And thrilled to be here. Our partnership with FFB, I think, has been a great example of that. Is there's just a lot of, I think, complementary skills and expertise that an organization like Atlas can bring and can benefit from working with Ben and his group. And so, the latest example of that we've had, and I think has been very successful so far, is a company called Bio. Some of you may know Vedere means to see in Italian. So this was a company that was purpose-built to go after some large unmet needs in ophthalmology. And we based this company around some technology out of the University of California, Berkeley. We really scoured the earth to look at many different cell and gene therapies and other approaches to try to think about ways that we could treat inherited retinal disorders, but in a genotype agnostic way. One of the challenges with doing gene replacement therapy, it can be very powerful, but for a very narrow set of patients. And so thinking about modalities and approaches and technologies where we could treat a much broader swath of patients, potentially get to those patients much later in disease, and think about clinical trials in a different way, where you're not running these very long, large trials to try to change the slope of the curve as you're losing photoreceptors. So this is an optogenetic approach that looks to impart visual acuity downstream of the photoreceptors into cells that survive decades after the photoreceptors are gone. Very new biology, new technology, very high risk, but if an approach like this works, the impact can be pretty extraordinary, we thought. So we were thrilled to create this company, bring in the technology, partner really from the outset with Ben and his team, who knew the PIs behind this work, understood the technology intimately, and really were big believers. And so they were able to bring their network, their deep expertise in the area, and Atlas was able to bring our expertise in building companies. So what we brought to the table was a CEO that could quickly build a team and an organization to translate this technology from academic work into something that now is on the path in IND-enabling studies, and hopefully will be getting tested in patients soon. So, that's our job, to sort of be on the cutting edge, find interesting technology, roll up our sleeves, and try to attract great people to run it, and then, be able to partner with groups like FFB to hopefully get it to patients as quickly as we can.
Any thoughts about, those are kind of blockbuster ideas, reinvigorating the retina, so to speak. But what about all of the other kinds of problems that, the more specific genetic therapies How do you see that, funding that?
Yeah, so Videre is one of a number of investments that we've made in the ophthalmology space, and I think it's just about finding the right tool for the right job. And so, this optogenetic approach was developed to try to treat a range of diseases. We also have a company that's developing non-viral means of delivering genes and other genetic therapies, a company called REGENXBIO, which we think can get to a point where you can actually redose these therapies, which, as we know, is an enormous challenge. At the same time, we're believers in the sort of workhorse of AAV, and we have a company called Affinia that is developing a next generation of AAVs with novel tropisms, the ability to get to different cell types, novel cell type specific promoters. So we think the more of these types of technologies and approaches, the better. And again, it's then about matching the right tool for the right indication. And the more of these we have, the better off patients will be.
That's great. Well, maybe we can do the next question over to Laura Manfre. Laura, since you work so closely with the patient community, and in our space, we've got over 300 genes identified. There's LCA 1, 2, 3, 4, all through the numbers. There's pure PH2. There's all kinds of things here. How do you deal with such a diverse community of people with all these different potential variants, and how do you get them interested in the science and potentially even in participating in clinical trials?
Yeah, good question. Thanks, Ben. And I think, as you know, our soapbox really is all about making sure that our patient community understands how important it is that they're heard, that our voice is part of the solution, right? We can't sit back and wait for treatments to kind of be handed to us and then just show up, right? So that's what as an organization we're really focused on. And it is confusing, it is complicated, it is a lot to understand. I mean, there's a ton of smart people here and it's a broad ecosystem. So it's not easy. We try, and of course we have a great partnership with the FFB where we lean on the FFB really to help with that science and with that, the research and the education. And then we focus our energies around really, again, disseminating that to the LCA community and other IRD space, but also providing, I think we're able to do a little more of the high touch warm fuzzies, right? Because the more we can empower people, the more we can keep giving them that hope as well, and just share stories about people living with this. And the more we have an opportunity also to keep connecting with them and educating them and informing them on where the science is, how fast it is progressing, what their role is in it, and why it's important that they participate in registries, they participate in natural history studies, all of these things. So that's my soapbox.
That's great. That's great. And I think maybe Richard, you could add on to that, given from your space where you're very your systems are uniquely positioned to help educate patients and physicians about what's going on with the science and the trials.
Sure, absolutely. What we focus on, the problem that we're trying to solve is how do you get the right information, condition-specific information, drug-specific information to the right patient at the right point in their care. And if you think about top 100 pharma and look at a drug 10 years ago, that drug had a target patient population of on average 700,000 patients. Today, a top 100 pharma drug on average has a target patient population of about 100,000 patients. And that's even narrower for the diseases that we're discussing on this call. And so when you think about it, it's how do you get that information in such a narrow patient population directly to that patient at the right point in time? And we've done that with technology, both at home through telemedicine and then in the office using educational kiosks and screens to do that. I think one of the key points to kind of piggyback on the last comment, over COVID, we saw a lot of what we call a diagnosis gap. So the data as of yesterday from IQVIA shows there were over 1 billion misdiagnosis visits last year. And those are defined as one diagnosis per patient per HCP. the top 5 specialties, as you guys can all imagine, number 1 was oncology, but number 3 on the list is ophthalmology in terms of that diagnosis gap. And so I think a lot of what we have to do now is understand how do we use technology to help these patients understand what diagnosis is and what the treatment options are so they can come in to the right healthcare provider to receive care.
That's great. That has a huge impact on the patient journey. We hear time and time again that it can take a really long time for a patient to get the right and proper diagnosis in our space. So thank you for that.
One question that is always interesting to me, and I'm going to direct this to Stéphane. As CEO, kind of in this case, in SparingVision, but others, kind of what keeps you up? This is what keeps you up at night. How are you positioning the company? How do you drive it clinically? How do you drive it economically? What is it that really excites you on one hand, but probably, like they say, keeps you up at night?
Well, I'm a very good sleeper. So having said that, as a CEO, you have a million day. Yeah, so you have to look at multiple parameters, being your non-clinical development, your manufacturing, your upcoming clinical trials, the natural history studies you have to do prior to enter into a human trial. You have to put together a team. You have to deal with your board. You have to deal with your investors. So if you start to be a little stressed by only one of these parameters, then you will no longer be a good sleeper. So I try to, in the day, work with all these parameters, deal with all these parameters, but I try to, forget everything so that I can have a good night of sleep and then start again in the morning. But if you start to worry about all these things, then you will never sleep. But at the end of the day, what is driving us is to go as fast as possible to bring novel drugs and innovation to patients. And every week that we are losing, every month that we are losing, every failure, at the end of the day, the first thing that comes to our mind when this is happening to us is we are losing an opportunity to have something that will benefit the patient.
Do you, and this maybe also we can ask Steve, when you swing for the fence, it's not like baseball. You don't really get 3 strikes very often. You get 1 strike. That's certain. How do you make sure you connect with that 1 strike?
Yeah, and especially when you have all those stakeholders sitting in the stadium that Stéphane referred to while that pitch is coming in. So, and you really want to connect on that pitch. So I think that gets back to some of the earlier comments of urgency, but really doing it the right way so that you don't have to go back. And I think that comes up in terms of in some ways, it's a very exciting time because the sciences move very quick. But as was mentioned earlier, you got to make sure that the production aspects are dealt with appropriately. The preclinical testing is dealt with appropriately, and all the way through to interactions with the FDA to come up with the right endpoints. And I think one of the challenging parts in connecting on those pitches is the science is ahead of even sometimes our understanding of natural history for one disease versus another, or how to best show that benefit that we all want to show. And one thing I kind of always go back to is, of all these stakeholders, the patient voice is key, because that's where you can really learn how to show that benefit in some ways. So really thinking outside of just visual acuity? What are the functional aspects or deficits that come? And one example, I learned of how important the continual voice of the patient and the patient's family is in drug development is our CLN2 Batten disease program, which is a program where our interest from REGENXBIO was initially as a CNS delivery of gene therapy to treat the CNS manifestations of CLN2 disease, which is a lysosomal storage disease that has significant CNS manifestations. Fortunately, there is a systemic enzyme replacement therapy for these children that has allowed patients to live longer. And what's happened is as the patients as these kids are living longer, they're unfortunately going blind. And that wasn't known before because the children were passing away earlier without enzyme replacement. So long story short, we have very active patient advocacy focus led by Vivian Fernandez. And we set up patient family discussion groups with our development team. And we were meeting with these families talking about how should we think about CNS endpoints or to show this benefit. And the families kept telling us, well, that's great that you want to develop something for the CNS manifestations, but can you find something to stop my child from losing vision? That's where our big issue is. And frankly, from those discussions, that's what allowed us to say, hey, that's really a big unmet, this is where we could really have a benefit. And we as a company also know a lot about local delivery for retinal disorders. And it wasn't too long after one of those discussions that independently we were speaking with you, Ben, about putting our heads together about CLN2 and how can we move forward in thinking about addressing that big unmet need. So I guess back to your question, Gene, I get more swings at the bat by having multiple shots on goal, but really optimizing each of those swings by really listening to all the stakeholders and particularly the families and the patients.
Great. Next question is for Kevin. I think it'd be great for you to comment on, we often get some pushback, these gene therapies and cell therapies are expensive, right? So could you comment on, risk reward, maybe even relative to other expensive therapies, and your thoughts on payers? How do we get these things reimbursed, and what are your thoughts on that?
Yeah, so to start on the risk reward topic, and maybe going back to Gene's prior question, and to torture the baseball analogy, given that the Red Sox are up to start this year. Honestly, if we're not swinging and missing sometimes, we're not swinging hard enough. That's kind of the way we look at the world. And if we're just focused on making contact, each time, you can't really do anything transformational that way. So we fully anticipate that we're going to take some very big swings, and sometimes they're not going to connect, and that's fine. That's part of our business model. But again, for the ones that do, you can have a really enormous impact. The pricing question, I think, ties into this, frankly, because the cost of all medicines, and especially these potential one-time treatments, is going to be a perennial issue, I think, no matter what the political environment is. In fact, it might be the one thing that both parties can actually agree on. And so we need to get very creative as an industry. And I think, frankly, we can't rely on payers to be the creative, innovative ones that are going to flex. I think our industry is going to have to be the one to do that. And you've already seen some of that with these novel payment models where both paying for performance, paying for these therapies over time. But, I have to say, when I look at what our industry has been able to accomplish, over the past decades, I have to be optimistic and think that, if we can't figure out if we can figure out how to permanently activate a gene with a single treatment in an allele-specific manner without ever cutting the DNA to treat a crippling haploinsufficiency disorder, I think we can figure out together how to get that paid for if it's really changing a patient's life. So I don't want to be too dismissive of the issue because it is an existential issue for our industry. But if we keep investing in breakthrough therapies and have the impact that I think we all think it can, I think it'll get figured out. But it does lead us all to move away from the me betters, the me toos, the reformulations, et cetera, which I think is a good thing for the industry.
Excellent. Yeah, I agree. If we can figure out gene therapy at this level, we should be able to figure out some of these business issues. So thank you for that. That's awesome. All right, Gene, you want to kick off the next one?
Well, there's a lot of work here on the panel. We talked a little bit about new vectors. I know both, again, Kevin with Videri 2 now, and Steve at REGENXBIO, and actually, the broader field. People are so excited about new vectors, but I see them as a lot of unknowns at the same time. How are y'all balancing that?
Yeah, it's it again gets back to that balanced aspect of urgency, moving quickly, but you have to show that these therapies are safe, of course. But if you look at the overall benefit risk, everything comes back to the benefit risk. We at REGENXBIO, it's interesting because our company was founded based on the overall platform that we have in terms of some of the most validated AAV vectors, so vectors like AAV8 and AAV9. So AAV9 is the vector used for the first systemic in vivo gene therapy program that's been approved, Zolgensma, for the treatment of spinal muscular atrophy. So one of our views is learning from all the different programs. So that was a program that we had out-licensed to Avexis, and which was then acquired by Novartis, for example. And we have over 20 different out-licensed programs, as well as our own internal programs. And one of the things we're very interested in is how the field can learn collectively. And each program is its own program, of course, in terms of the promoter, the transgene, the context of the disease that you're treating. But it's really an exciting time for the whole field to learn across the different AAV vectors. So I've been talking about things outside the eye, of course, also in the eye. There's subretinal delivery, which has been the most utilized for a lot of the IRDs and non-IRD indications. People are looking at different routes of administration, like suprachoroidal delivery. So I think there's still a lot to be learned about all of these vectors and routes of administration. But to Kevin's point, you gotta swing for the fences in some cases, but along the way you gotta really track the spin of the ball. Boy, we're really torturing the baseball analogy.
Yeah,
football.
The only other thing I would add to that is it's another reason for everyone on this call to be optimistic, right? Because the toxicity concerns around AAV and vector copy number aren't going anywhere anytime soon. And so I think you're going to continue to see, whether it's the workhorse, REGENX, AAVs that came out of the Wilson lab or next generation vectors, you're going to continue to see the eye as kind of a beachhead for use of these new technologies due to the relative ease of getting to the target tissue, lower VCN that is required. So I think this is where you will see sort of the front lines of innovation in terms of finding newer, safer, better tropisms, more tropic vectors is really in this space.
I have a follow-up question to Wiley. Okay, we have these wonderful new vectors. They hit the exact cells at the exact place at the right time and seem to be exciting, but we just don't have any history with it. How does the FDA look at new things?
So we don't particularly care whether it's new or old. We care whether it works. So if you show the product works in people, we'll approve the product, new or old.
Do you have any concern? I mean, now we have patients that have AAV for years, AAV2, but what about something new? Do you long-term safety? How do you manage that?
Something always has to be first. There is nothing that we approve that we know everything about at that point in time. We try to continue to collect information after approval to be able to help answer those questions. But we don't want to wait till we know everything about the product and people have missed out on 30 years of using the product. So we make an educated guess, approve the product, and follow it.
Makes sense. Stéphane, I think you had maybe a comment on delivery as well, and then Tarran, maybe you could speak a little bit to the role of manufacturing and purity as it relates to safety of drug product. So Stéphane first.
Yeah, thank you, Ben. So yeah, it's not too much on the vector, but more on the delivery. I mean, so far we've been looking at in the eye mostly, subretinal way to go after, the photoreceptors, and there is nothing more efficient today. But if we want the field to move to more prevalent disease, it's fair to say that we will need different routes of administration. And I know that many people are working on it, and definitely this is very high in the agenda of SparingVision. We need to find a novel way to distribute and transduce the product effectively in the photoreceptor, because it's okay to go this way or this route for rare disease and ultra rare disease, but if you want to move again into more prevalent disease, that won't fly.
Yeah, makes sense. And Tarran, do you have a comment?
Definitely, I'm very excited about the new vector space with some really great technologies coming out of really cool companies like Dino Therapeutics. So I'm very optimistic about the new vector space and how it's going to impact delivery and having better tropism, having lower doses that can improve safety. But in terms of the manufacturing, there's going to be some extra work that we need to do in terms of purifying those new vectors, because there aren't going to be off-the-shelf products to purify. So different resins will have to be created, and also accompanying analytics to be able to measure the purity profile of these new vectors and as you manufacture them. So I think that it's possible, just some additional work will have to be done in order to have the right tools in place to scale up, but also to remove the impurities and to capture and to remove the impurities in the manufacturing process, and the new analytics to measure.
Great, thank you.
One thought is, what does the future look like? Maybe we just could kind of go through each of the folks, maybe again in alphabetical order, and give us your sense of what you see for the future, what you would like for the future, something like that for the next few minutes.
Sure. Thanks, Gene. I think that, I think what happened in COVID has accelerated the use of technology in healthcare at least by 5 years. And it has done that, as we've seen in development process, but in our case, it's done it in terms of adoption of healthcare providers, how we interact with our patients, how we take care of them, and how we manage chronic conditions and seeing patients follow up. In some of the verticals we work in, like rheumatology, for example, over 50% of patients were seen over telemedicine during the pandemic. And if we look, as we watch some of these changes, now that's come back down. Rheumatology now is about 13% are seen over telemedicine. But some specialties have maintained that digital connection with patients. In psychiatry, if you look today, over 60% of patients are still seen on telemedicine, not in office. I think the other big trend that we noticed is that we measure diagnosis and prescription data in each of the offices that we're in across the country. New to brand RX, so starting a patient on a new prescription over telemedicine happened for the first time during this pandemic. And in certain specialties like dermatology, dermatologists felt very comfortable starting a new patient on a new to brand prescription electronically without seeing a patient. Now that's changed a bit as the pandemic has come back. As we look now, we look at traffic, foot traffic in doctors' offices, and that foot traffic has come back to pre-pandemic levels. In certain specialties has exceeded it. But what we're seeing is that in certain specialties, doctors are still using telemedicine to augment the patient visit. Doctors do feel comfortable writing and starting patients on new therapeutics over telemedicine. And I think the final big change that we're going to see is the use of field force. Due to COVID restrictions, a lot of pharmaceutical companies were not able to have their field representatives in the office. And that's important because educating the doctors is an important part of getting new drug information to patients. I think as we come out of this, we'll see technology really change the way that pharmaceutical companies interact with doctors and the way that those representatives are able to engage with practices, with physicians, and really start that cascade of educating patients.
Cool. Kevin.
Yeah, so maybe I'll jump to the other end of the spectrum. Could probably list a dozen different areas of emerging technology that we're quite enthusiastic about. I think, as it relates to advances in the eye, particularly keen on watching the confluence of what we've been talking about a lot on this call, next generation cell and gene therapies with the emerging biology around regenerative medicine. And so, I mentioned before the efforts at a company like Awdeh to essentially do an end around of photoreceptors and try to get to the downstream cells. But wouldn't it be wonderful if we could, replace degenerating photoreceptors with healthy new rods and cones? And so whether it's next generation cell therapies, where you're seeing a lot of progress and moving away from just making RPEs to be able to make bona fide photoreceptors, to exciting areas like doing in situ transdifferentiation of retinal stem cells. I think there's a lot of exciting science to tap into there. It will be quite applicable to a variety of different disorders in this space.
Great. Stéphane, I think you're next.
Yeah, thank you, Ben. So yes, we believe that the future obviously is in mutation, agnostic gene therapy when it comes to gene therapy, as it would be very difficult to address in an economical manner all mutation specific therapies. Unfortunately, some mutation represent only a very small subset of patients, and it would be difficult to find any ROI that could justify an investment. Then when it comes to other technologies, we believe that some of the gene engineering tools, such as gene regulation, should be more widely in the space, like they are starting to be in the brain, for example. And last but not least, we are big believer in vivo programming, and this is just the beginning, but this is definitely a place where we will be investing in the future. It's not going to be for tomorrow, but if in 15 or 20 years down the road we could turn any kind of important retinal cells into a cone or into another, critical for vision cell, that will be a fantastic avenue.
Great. Wiley.
So long-term goal is easy for me. I'd like to see cures. Treatments are fine, but I'd like to see cures. On a shorter term basis, I would like to try and see companies spend more effort learning from a particular trial before they do the next trial. What happens now is we get, and I do understand why, the plan for the next 6 steps. And the plan for the next 6 steps, if you're telling me what it is now, you're not planning on learning from the steps in between. And I think it's critical to be most efficient by learning from each individual step. And the thing that probably bothers me the most is to have these what are called expansion trials. You just now go and treat the next 10 patients, or the next 15 patients, or the next 100 patients. And that's not, that's wasting basically potentially valuable resources, meaning the patients, in a way that we could learn more from what happened.
Very interesting. Thank you. Laura, you're up next.
Yeah, so I'm going to second what Wiley said. We want to see more cures. Having said that, I think it's also really, really important that saving any amount of vision for any amount of time just makes a huge difference in the lives of individuals who are facing blindness and their families and all of that. So any amount of vision that can be saved with any treatment is very, very significant to those living with rare disease. Our last family conference that we had, we had both a family who was there with maybe a 7 or 8 year old who still did not have a genetic diagnosis, but they thought it was LCA and were pursuing diagnosis. And at that very same conference, there was a mom who came who I think she had a son who was maybe 3 years old. And in the first couple of years of life, he was diagnosed. He was diagnosed with RP65 and he received Luxturna. So that's what we want to see more of, right? Quick diagnosis, understanding. They found Sofia Sees Hope. We were able to welcome them in the community. They could share their stories. They found the FFB, and they were able to receive treatment. So more of that.
Amen to that. That's a great story. Steve.
Yeah, so in many cases, the science is ahead of some other aspects that we've talked about here. So I guess what I hope to see in the future is that some of those other rate limiters catch up, like manufacturing capacity, efficiency, quality, our understanding of these rare diseases, each of them in terms of understanding the natural history and evolution and how to show benefit for these patients. And then third, how to pay for them. So payment models that allow for what we all want, which is to get these treatments after we show them to be safe and effective to the patients.
Great, thank you. Tarran.
Yeah, just to, I guess, echo a little bit on what Steve said regarding manufacturing. Pre-COVID, there was already a capacity crunch in manufacturing, and COVID just escalated that issue. And I think that the ability to democratize manufacturing so that we can get more shots on goal to get more potential solutions to patients through the clinical trial process is going to be super important. And capacity is not the only way. Adding more capacity in manufacturing is not the only way to do that. I think it's finding better solutions, bioengineering, but also digital engineering to allow more options to be tested in patients and to get therapies. So I think democratizing accessibility to manufacturing is super important. It's really the future here, because adding capacity isn't the answer. It's finding better ways of manufacturing.
Yeah, that's great. I think that makes a lot of sense, because we don't need big batches for most of the stuff we're talking about. So it's really about that efficiency and the quality. So thank you for that. All right, we're getting close to the end, but Gene, do you want to comment or?
Well, I have a question for you, Ben. I mean, you're sitting at the kind of the top of the heap on one of these, one of the most important organizations in our ecosystem. What are you trying to? What do you see as the future, and what would you like to see under your watch?
Yeah, look, I think we're trying to solve all these things at the same time, which is it's a big goal. But I often think that we get driven into decisions I call sucker's choices, and so I'm much more of a yes and kind of guy, and not a yes one way or the other. So I think we can actually do it all if we are smart about it. So the way I see it is these gene replacement therapies, where there's validated biology, we have to take the rifle shots now and get those done. We have to take the big swings on these gene agnostic approaches to cover as much genetic territory as possible. We have to do that. We have to also work on regenerative medicine, because there's a lot of promise there, and to actually restore structure is a huge issue. So we basically have to do it all. So I imagine the future where, sort of like Stéphane was saying, we'll be having gene editing of cells that get put back in the eye to do remarkable things. And I think we have a chance to do it all if we have enough funding to do it, because the science is pretty much getting there.
Thanks. I just personally, I do think that the suffering that we know, we see as physicians and parents, and really is the driver. I sometimes tell my team on different times, and just think of that person sitting right next to you, not saying anything, just watching you, saying, why aren't you working a little harder? Why aren't you being a little smarter? Why aren't you helping me? And I think that's a huge motivator, and so I want to help those people. And I know that's why we're all here. Thank you, Ben.
Yeah, thanks, Gene. Thanks, everyone, for joining. I think it was a great discussion. Really, thank you, Gene, Richard, Kevin, Stéphane, Wiley, Laura, Steve, and Tarran. All of you just really contributed to a great conversation here. Also, a big thanks to our partners to help make it happen. We have support from ICS, REGENXBIO, Biogen, and Charles River. So really appreciate that support. So next up, we have some closing remarks from Adrian Graves, who's on the board of directors for both the foundation and the fund. But to get there, you have to close out of this session, and then select the Investing in Cures Summit closing remarks and select join session. So again, thanks for joining us for Investing in Cures. Got one more little segment to go, and thanks for your participation.
Thanks.
Bye.